So the overall problem in our field is that we need to develop diagnostic methods to identify Alzheimer's disease in our patients, and we need to develop improved treatments that will slow the progression of Alzheimer's disease. 20 years ago, the field of immunotherapy began when it was discovered that antibodies could remove amyloid plaques from the brains of mice. And the pharmaceutical industry and researchers began to think about developing treatments for humans using monoclonal antibodies. But at that time, we had no biomarkers to identify Alzheimer's disease in people or to measure the progression of the disease itself. We only had clinical measures that could measure cognition and activities of daily functioning. I proposed a grant called the Alzheimer's Disease Neuroimaging Initiative, which was funded by the National Institute of Health in 2004. It's one of the largest funded projects on Alzheimer's disease in the world. The overall goal of the Alzheimer's Disease Neuroimaging Initiative, which is called ADNI, is to validate biomarkers for Alzheimer's disease clinical trials. And over the many years, we've enrolled over 2,000 participants at 60 of our participating centers. The participants are completely normal people. They are people with mild cognitive impairment. And we have, of course, some people who have dementia, which we clinically diagnose as Alzheimer's disease. And then on top of the enrollment, we have a group of assessments, including activities of daily living, cognitive functioning, and neuropsychological tests. And then we have a series of biomarkers. Everybody has had blood drawn at every visit. We've been doing MRIs on people for many years. We began with fluorodeoxyglucose or FDG PET scans to measure glucose metabolism in the brain. And then when the amyloid PET tracers were developed, we initially did a study with Chet Mathis using the Pittsburgh compound PIB. And then we switched over to using fluorobetapir, which was provided by a company called Avid. And ever since then, all of our participants have been having amyloid imaging every other year. And then when the tau tracer for tau-superior was introduced, we started doing tau imaging. And then as other tracers were developed by other companies, we've been using those tracers as well. So all of our participants are extensively phenotyped. And I should also mention that many of our participants have had lumbar punctures to obtain cerebrospinal fluid for measurements of amyloid beta and tau. I enrolled in the project as a participant, as a normal healthy control, because I met the age requirement. And I've, for the last 20 years, I've been having visits every year or every other year, and with MRI scans, PET scans, lumbar punctures, all of that. And you learn a lot from doing your own trial. And a unique thing about ADNI, several other unique factors. First of all, it's a partnership between a government-funded project and industry, and we have contributions from just about all of the major pharmaceutical companies and a lot of other smaller companies involved with diagnostics and biotech companies. We call that the Private Partner Scientific Board, which has contributed 20 or 30% of the funding over the years. And we also have made all of our data available on the internet. So any scientist in the world can go to the ADNI website at ADNI USC Loney ADNI and obtain a username and password and sign a data use agreement, and they can get access to all of the data. That is all of the MRIs, all of the PET scans, all of the genetic data, all of the blood test data. All the data that we have is available to any scientist in the world who wants to request it. And that has led to the publication of over 4,000 papers in the last 20 years from this project. It's, as far as I know, in the world of Alzheimer's disease, no other project has had so many publications from its data. And that's because of our open release policy.